photosynthesis, there's not even light involved. It's not even something happening at the membrane, but please stay. There are some analogies to photosystem too, and maybe that might be interesting for you. So I have to bring you up, of course, to the carbon cycle, the global carbon cycle, and of course that was what is well known and of course well studied is this part, carbon dioxide being reduced to organic matter and then respired. What we speak about here is the aerobic oxic world. That means everything in the presence of dioxygen. What is substantially less well known and less appreciated is that similar things, of course, like this carbon cycle happen under anoxic conditions in the absence of dioxygen. So there's a lot of life that converts carbon dioxide into organic matter, and one pathway is especially interesting called acetogenesis. It's not only acetogenesis, but it's of course also other processes like methanogenesis that convert carbon dioxide to methane, but also from organic matter. And I tell you about these pathways because there's one central enzyme that you find about in all of these anoxic pathways, which is called carbon monoxide dehydrogenase. So that is the one acronym that you have to keep. CODH, that stands for carbon monoxide dehydrogenase. I try to avoid acronyms otherwise, but this one is the one I need. So 4 times you have it in a reoxidation of organic matter, the last step when carbon monoxide is oxidized to carbon dioxide. Organisms use it for reducing carbon dioxide to carbon monoxide to make this acetogenesis. Organisms use it for reducing carbon dioxide to methane, but also to converting organic matter to methane. So if you're an anaerobic organism, you probably would want to have a carbon monoxide dehydrogenase. There was just another carbon monoxide dehydrogenase shortly mentioned. I just want to also mention it shortly. There's one carbon monoxide dehydrogenase that occurs in the aerobic world. It's an oxygen-stable protein. It contains copper and molybdenum. The one that I will speak about is you just find in anaerobic world, just with organisms living in the absence of the oxygen. There's a simple reason: the protein is oxygen sensitive. So that's the one that contains nickel and iron, and that is the focus of my talk here. So why could that be interesting? First, with an aspect of reducing carbon dioxide. So this pathway, acetogenesis, is estimated to be responsible for about fixing up to 20% of the global carbon dioxide. So it's not only about photosynthesis when you speak about fixing carbon dioxide. There are other pathways, just less well known. And this pathway was discovered by Harlan Wood and Lars Ljungdahl. They are the pioneers of this pathway. And they discovered that it's a pathway in which 2 carbon dioxide molecules enter, both become reduced, one by 6 electrons down to a methyl group, which is equivalent to methanol, and one by 2 electrons forming carbon monoxide. And this enzyme that does this reduction is this carbon monoxide dehydrogenase. As you can see here, it just does both reactions. Now, it can oxidize carbon monoxide to carbon dioxide, but it can also here in this pathway. It's important for reducing CO2 to CO, which is then actually coupled when it's produced to this methyl group and gives acetyl-CoA in the end, so activated acetic acid. So this pathway is, I think, interesting for many reasons. One of the reasons is probably the way how, at the very early age of evolution, CO2 was fixed. So it's probably, as people estimate, it's about the oldest CO2 autotrophic pathway of all. So it's a very recent paper here on the phylogeny of carbon monoxide dehydrogenases, and they made the, daunting talk to take 6,000 or more than 6,400 completed genomes of archaea and bacteria, compared them, found that carbon monoxide dehydrogenases, or genes for carbon monoxide dehydrogenases, are so widely spread that you have to say that Luca, the last universal common ancestor that lived around, estimated 3.5 billion years ago, most likely had a carbon monoxide dehydrogenase. So you find it in a lot of different archaea, you find it in bacteria, you can't resolve it here. These are too many sequences. It's just showing you that now all these genomes that we are having give us insight on what probably was there in the early life, and carbon monoxide dehydrogenases were there. That is, I have just taken it from a very recent paper, but that of course came to no surprise because there are evolutionary biologists like here Bill Martin and Michael Russell, they are saying for a long time that probably when life evolved, it evolved in something like these black smokers. And the first pathways that evolved over there, they were called Acetyl Genesis, living from carbon dioxide, making organic matter from it, and Methanogenesis. So these two pathways are probably about the oldest pathways that you can find. And in both of them, you have carbon monoxide dehydrogenases. Okay, so I hope I could give you a little bit of bearance with me that you like to hear more about this carbon monoxide dehydrogenases. The chemistry of them, in principle, when you look at it, of CO oxidation is not so complicated. CO oxidation is, of course, well known, and one of the most famous reactions here is the water-gas shift reaction. Still, I would like to shortly mention it because it tells you the basic parameters, the basic requirements that an enzyme has to meet when it wants to oxidize carbon monoxide. So the water-gas shift reaction here is abbreviated Metal4M. It binds the carbon monoxide, it activates the water so that it becomes more nucleophilic. You get a hydroxyl ligand. The hydroxy attacks the carbon monoxide once it is bound to the metal, not before. You form metal carboxylate, which undergoes a beta hydride shift. So this proton with 2 electrons is going to the metal. You eliminate carbon dioxide, you have a metal hydride, which then typically becomes protonated, and you liberate dihydrogen. That is what happened in the water-gas shift reaction. That's not what happens in the enzyme. The enzyme is keeping the protons and the electrons separate because the organism wants to live from the electrons. Actually, the pathway has another important parallel to the chemical industry, and that's the Monsanto acetic acid process. So this reaction that you've just seen, this condensation reaction of a methyl cation, carbon monoxide, that is actually similar to the acetic acid production in the Monsanto and Cativa processes, which are using rhodium and iridium to make this type of condensation reaction. These are two additional reasons, I think, to study this pathway and the enzymes, because the enzymes are not doing it with rhodium and iridium, the enzymes are doing it with nickel and iron, and that, of course, is much cheaper than having rhodium and iridium. So these two principal catalysts of this pathway, this acetyl-CoA pathway, where you take carbon monoxide or carbon dioxide and make acetic acid from it, they're two that have been studied most intensively. And one thing is for the chemistry, another thing is for the evolution, and the third that is most attractive to me, they contain unusual and unique metal centers. That is the active site metal center of this enzyme that can convert carbon dioxide to carbon monoxide. And you can see here it contains nickel in an iron-sulfur cluster and an additional iron. The enzyme that converts a methyl cation, carbon monoxide, and CoA to acetyl-CoA makes this condensation reaction, contains 2 nickel ions next to a 4 iron 4 sulfur cluster. These metal sites have never been seen in any other enzyme. They are absolutely unique for this 2 enzymes reacting on carbon oxides, carbon monoxide, carbon dioxide. So for the talk, I will focus completely on this left side here because for several reasons. One is that it has some parallels to the photosystem too. But first, before I go to the structure of it, very shortly about this organism. I showed you that these genes for carbon monoxide dehydrogenases converting carbon dioxide, carbon monoxide are widely distributed. And we are working with a special organism called carboxydothermos hydrogenoformans. And this organism is indeed an expert in using carbon monoxide, which is nicely shown when the genome was published. They found 5 different genes encoding carbon monoxide dehydrogenases. And this multiple already indicates that it uses this enzyme for different purposes, under different conditions, under different regulatory conditions. And we have one that is obviously important for energy conservation. It's coupled to an hydrogenase, because what this organism is doing, when it lives with carbon monoxide, it takes the carbon monoxide and it produces hydrogen. That's why it's called hydrogenoformans. It just blows off hydrogen in the air. The second one is probably important for NADPH generation. The third one is important for carbon dioxide fixation. That is part of this genome with acetyl-CoA synthase. There's one for oxidative stress response, and the fifth one that we don't know. But you can see this organism is a specialist on using carbon monoxide for different purposes. So here I will focus on one carbon monoxide dehydrogenase, which is this carbon monoxide dehydrogenase 2. And there's a simple reason. It gives the best diffracting crystals. I'm a crystallographer. So that is the structure of this enzyme, this carbon monoxide dehydrogenase. That's why there's always a 2. It's a number 2. The other ones are typically sharing sequence identities of 60 to 70, 80%. So they will look the same, and the ones that we studied already look the same, really. So it's a prototype. It has 5 metal clusters, always iron sulfur. So no manganese, no oxygen here. You have standard type cubane 4 iron 4 sulfur clusters called D and B. They were named before after their spectroscopic signatures. And then at the outside here of this V-shaped electron transfer chain, here at the outside, you have a cluster called cluster C that contains nickel, iron, and sulfur. So active site is here outside, and this is just for electron transfer. And you see the typical distances that we are finding when there's a rapid electron transfer. Everything below 15 angstrom here, 10, 11 angstrom between shortest iron-iron distances. So I will focus on these guys here on the outside, on this C cluster, where the principal reaction, this carbon dioxide reduction or CO oxidation, is happening. So this metal cluster is unusual compared to other iron-sulfur clusters in that it is known to have at least 4 different oxidation states. Typically, for iron 4 sulfur clusters have 2 oxidation states under physiological conditions. If you drive it out of the physiological, you sometimes observe 3 oxidation states. This one has 4. It has an oxidized inactive and diamagnetic. Then comes a paramagnetic state that is capable of oxidizing carbon monoxide, that is transformed into a 2-electron more reduced one, also again spin 1/2, paramagnetic. This reduction occurs with a midpoint potential of about minus 530 millivolt, which is more or less exactly the same as the midpoint potential between carbon monoxide and carbon dioxide. That fits very nicely for a catalyst that is 100% reversible. No? The midpoint potential of converting C-Red1 to C-Red2, which is 2 electron more reduced, is the same as for the substrates, and this C-Red2 state then can nicely take up carbon dioxide, reduce it to carbon monoxide, and then the enzyme would go to the C-Red1 state. So it's perfectly adapted by its midpoint potential for the reaction it's catalyzing to be reversible. To go back, otherwise it delivers 1 electron. C-Red2 goes to a C-in state, which is integer spin state, gives another electron, and then returns back. This happens with a kcat of up to 40,000 per second when you drive it in the CO oxidation direction. The enzyme is very much adapted to really capture carbon monoxide. We just really recently picked up an enzyme, this carbon monoxide dehydrogenase 4, which has a nanomolar Km value for carbon monoxide. So this CODH4 at all temperatures and CODH2 at high temperature have a kcat over Km of 10 to 8 to 10 to the 9 per second and molar. These are important data for us because it tells us these are perfect enzymes, they're perfect catalysts. Every substrate binding is converted because they're just limited by diffusion of carbon monoxide. And you can actually, for the CODH4, we even calculated the diffusion rate of carbon monoxide in solution to a buried active site for different temperatures, and the enzyme completely matches with this kcat to Km, the diffusion rate of CO in solution. So one of the few, actually relatively few, perfect catalysts. There are not so many, it's about 10. It's not so many enzymes are really perfect. So that is the active site. So I showed you already a little bit. It's a nickel-iron active site. And for good reasons, we can focus here on just for the beginning, at least on this right part here of the active site, the nickel ion and 1 iron. This 1 iron has a hydroxyl ligand, and you see something here which looks more like a 4 iron 4 sulfur cluster. And I will walk you slowly through the geometry of this cluster, because of course the parallels come here in the geometry. When we describe the cluster, you would say you have here a heterocubane with 1 nickel and 3 irons and with 4 sulfurs, and you have an additional iron in exo. I know you don't call it in exo, you call it dangler. No? I know that, but I come to that in a minute. But chemists call that an exo, they told me. I'm a principal, I study chemistry, but it doesn't matter. But, so that's why I call it in exo. It sits here outside. It's not part of the cluster, it sits outside. When you compare it to the normal 4-iron 4-sulfur cluster, you obviously see, yeah, well, this left part here is similar to 4-iron 4-sulfur cluster. The right part is not any more similar. There you break it apart. That's why people, pioneering people here like Dick Holm, they called it bridged iron-sulfur clusters. It's part of a series of different bridged iron-sulfur clusters, and of course the most famous bridged iron-sulfur clusters are the P cluster and the iron-molybdenum cofactor of nitrogenase. So there are some cases where an iron-sulfur cluster is taken and some other unit is in a way fused to it. And actually, a side aspect, what we found also recently, another bridged iron-sulfur cluster, which is a double cubane cluster we've just published, came just recently out. But so it's a bridged iron-sulfur cluster, something like an unusual 4-iron 4-sulfur cluster. And of course, ta-ta, now that comes especially for Jim, because that is something that we have in common. Robert Huber, when Jim's structure was published, recognized that the two structures have something in common. They are the only known biological metal clusters where a heterocubane is formed. Heterocubanes actually also don't, you find a little bit, maybe in nitrogenase, you could speak if it's a part of a heterocubane. But here's a heterocubane with an additional metal in exo, and that exactly, of course, is here. No? Just explain, just put for exo dangler, and you see the, of course, very clear similarities between these two metal clusters. And Jim compared it in this very recent paper in Nature Plants. He also superimposed, and you could see the clear similarities between these two clusters. There are some, of course, differences. And of course, I have to also point out where the difference. Of course, the metals are different, and of course, the ligands are different. And there's, of course, a small difference in the geometry, which is, for this nickel-iron enzyme, relatively important. Like what you can see here is that this cubane is already broken up. No? There is no direct bond between nickel and this sulfur, so it's a different geometry than the calcium. So that is a bit different because we have a nickel, which is here nickel 2+, a metal that has a 3D 8 electron configuration that likes to have a square planar coordination. And that is an important point. Why this is important and makes it difficult, I will just like to briefly allude with strategies that have so far been used to synthesize a cluster which is similar. You can imagine this is a very efficient catalyst for carbon dioxide reduction. It doesn't need any overpotential, it's rapidly turning over, it just needs nickel and iron. So it's an interesting starting compound, of course, for cheap CO2 reduction, and that's why some efforts have been undertaken to model the geometry of this cluster. There are two main groups that worked on it. Was Dick Holm and Katsuyuki Tatsumi, and they came relatively close, but described also what are the challenges. Why is it difficult to model this cluster? So it's not so difficult actually to get a heterocubane where nickel is inserted in an iron-sulfur cluster. That is possible, and that was known before. So nickel, iron-sulfur cubanes, heterocubanes are possible. But there now came the challenges. One challenge was to give the nickel a square planar coordination. And that was only possible here in the Dick Holmes group when they used a ligand that has a relatively strong in-plane ligand field that actually made this square planar. And then actually the bond between the nickel and the sulfur, like in the iron-sulfur cluster here, is broken. And the nickel goes off and it forms a square planar coordination. A challenge that has so far not been overcome, at least what I know from the literature, is this additional iron. There's no heterocubane with a dangler or with an exo-iron known for this enzyme. So to model this was so far impossible. What also was impossible to actually decap in a way the ligand here. Again, all the models have saturated coordination. Of course, this way they are not capable of any CO2 reduction. But that's, as far as I know, the most similar right now for modeling. So with this, I come back to the structure. So just that was a short detour, and I come back to the slide. So when I determined this structure, then a sentence of my former boss came into my mind. So you have to be very careful what you tell your PhD students. Probably some of the sentences they get ingrained, they stay in the brain, they will never go out probably. And one that I got stuck with was, I don't like naked structures. That is the original sentence of Robert Hoover that he said sometimes, and of course it's easy to understand. Naked structures is something that doesn't tell you much. It's a structure, but you don't have an inhibitor which is similar to a substrate or no substrate bound. You don't learn so very much about it. That's why we said, okay, naked structures are not good. Let's try something else. And what we tried was catalysis in crystals. The nice thing about protein crystals, and of course this with the X-fill is clear already, the nice thing is that enzymes in crystals often, not always, but often can still turn over. And we used this here. We gave the crystals, we incubated crystals with carbon dioxide. We incubated them with a strong reducing agent. We used titanium citrate. You have to go below this minus 530 millivolts. You need a strong reducing agent. And what happened then is that we get this cluster here. We have again the nickel and the iron, but when we let it turn over with carbon dioxide and then freeze trap it, we see something that obviously has 3 atoms. So that is not the original electron density that I'm showing here. That is right now the highest resolved structure that we are having, and I'm very happy, although it is very just below the 1 angstrom, there's a 0 in front of it. I'm very proud of the 0. So it's 0.97. If 1 angstrom would be the same, but it's nice to be below the 1 angstrom. Why is it here important? Why could it help us? What you can see here, the carbon dioxide is obviously not linear anymore. And this geometry of carbon dioxide, when it is bound, is very important because it tells you in which state the carbon dioxide is. So we shortly walk to it. So what we have here, carbon dioxide in this turnover condition is bound to the nickel. It forms a nickel-carbon bond, and here you have the 2 oxygens, and obviously they are bent. You have a carbon-oxygen angle of less than 180 degrees, obviously. That is important because whenever you activate carbon dioxide, you have to bend it. This way, when you bend it, you stabilize the LUMO, the less occupied molecular orbital, lowest unoccupied molecular orbital at the carbon. That is a place where you want to inject electrons, and you actually stabilize also the HOMO at the oxygens, just at actually the electron lone pairs of the oxygen. So that tells you, this molecular orbital picture tells you where you want to put the electrons in. The nucleophile should attack here at the carbon of this bent carbon dioxide, and that is where our nickel is interacting. And at the oxygens, you have hydrogen bonding interaction, and you, of course, at one of the oxygens, you have this iron. So you focus here on the nickel iron, they are doing the job. Well, that the carbon dioxide is bent, we've seen already at lower resolution. For that, this 1 angstrom resolution was not important. What was important for that was that we could refine the distances between carbon-oxygen, so bond distances. We can use actually methods that are typically used for small molecule crystallography. You make a complete least square matrix refinement, and then you invert the matrix, and this way you even get for each individual bond an estimated uncertainty. So like actually small molecule crystallography, that helps a lot in determining how well do I really know the bond lengths, because that is a problem in protein crystallography. You typically use restraints, you bias your refinement. Here is unbiased, you don't need any restraints anymore. And you see that the bond length here is about 1.3 angstrom for both oxygens. What you also see is the angle. And this angle is very important because the angle of 117 degrees immediately tells you that the carbon dioxide took up 2 electrons. So there are 2 electrons already inserted in this carbon dioxide molecule. It's a small OCO angle. 133 you would expect for a single electron reduction. 117 is what you expect for model calculations for a 2 electron reduction. The same is actually told by the long oxygen-carbon distances. They also tell you that the double bond character is lost to some good extent. So that was very interesting. The other thing that was for us completely surprising, and I can't really give a good explanation for it, is a very short nickel-carbon bond of about 1.8 angstrom. And when we saw that, we said, well, that is probably due to substantial pi back donation, so some strong interaction between the carbon dioxide and the nickel. And then after one of the talks that I gave, then Tom Rauchfuß, an inorganic chemist, came to me and said, Holger, you may call it substantial pi back bonding, I would call it the nickel carbane. So there's a true nickel-carbon double bond character here, and that would actually even potentially explain why this is a really activated state, because it weakens the carbon-oxygen interaction. You probably need just a protonation, and now you would split the carbon-oxygen bond here at this point. Surprisingly, that was our hope, that carbon dioxide is not really very much distorted. Actually, it's perfectly perpendicular to the plane of nickel and the sulfurs, and forms a plane here with nickel and the irons. It's very slightly distorted. That is one of the keys of very high resolution crystal structures of substrates. You often find that they are distorted so that in additional activation. That is not the case here. You don't see any additional distortion in this bound carbon dioxide except for this 2-electron reduction. So that was one thing that, of course, told us quite a lot about how carbon dioxide could be reduced, but we approached other aspects, and that was trying alternative substrates. So this enzyme is not only turning over carbon monoxide, but it also turns over isonitriles. So isonitriles are specifically binding to the state where carbon monoxide can bind, and they are also oxidized. It's a very slow substrate, but it's turned over to an isocyanate. The enzyme has several inhibitors, so you're looking at your inhibitors like ammonia and asking yourself where they are binding. We have inhibitors here that are specific for each oxidation state. Maybe not so different to the oxygen-evolving complex. You have an inhibitor sulfide, which binds to the oxidized state. Cyanide, isoelectronic to carbon monoxide, binds to the state where carbon monoxide is turned over. Cyanate, isoelectronic to carbon dioxide, is binding to and inhibiting the state where carbon dioxide is binding. And so we studied all the crystal structures, or we studied them at relatively high resolution. Here, this isonitrile, we can see in a state where it's already turned over, an isocyanate is formed. Cyanide is interacting here at the nickel. It binds at the nickel and binds so tightly that carbon monoxide can't replace it easily. And cyanate is actually just bound exactly like carbon dioxide. Here you have again the nickel and the iron, and you see the NCO angle is exactly like the OCO angle. It binds at the place of carbon dioxide and just stays there, is slowly converted to cyanide. So what do we learn from all these different crystal structures? Here they are superimposed. Left-hand side, you have the cluster. Here in the middle sits the nickel, and here you see the different ligands. So one thing that you're seeing, all of them are interacting here with the nickel, and the nickel hardly goes off its plane. The second thing, you find a clustering of oxygen interaction at residues which are in hydrogen bonding distance. Here's a histidine residue in hydrogen bonding distance, and here's a lysine residue in hydrogen bonding distance, and they are probably there just to give a direction. Where should a ligand bind, and where can it be stabilized just by hydrogen bonding interaction? So these are hot spots from the second coordination sphere around the metal to distinguish or determine where will be a ligand stabilized. We have two geometries around the nickel. One, the dominant seems to be, of course, here square planar geometry, but there's one here with this isocyanate. Here's a carbon, here's a nitrogen. There, the nickel goes a bit off, and it's partially tetrahedral. It's a distorted tetrahedral coordination. So from this, we know where ligands are binding. We get an idea, which I'll tell later about the mechanism. But there are several open questions, and one of the open questions is, why do we need the cluster? Everything I told you happened at nickel and iron, and we still have this cluster around. And of course, a similar question comes with the OEC in a way. There you know that the oxidation equivalents are stored. Here it's unknown. When we superimpose carbon dioxide bound with a non-carbon dioxide bound, when the nickel has a T-shaped coordination, we see there's nothing happening in the geometry. It's practically the same geometry if it's T-shaped, three-fold coordinated, or four-fold coordinated square planar. That tells us this is type of surface chemistry. Now, everything happens here at this diet, and the cluster doesn't rearrange. That's probably important because the cluster turns over with about 40,000 per second. If it would have large rearrangements, it would probably have a large reorganization energy and would slow down catalysis. But this is obviously something like a surface type catalysis. It doesn't need large rearrangements. Okay.
No, I'm just sorry, I'm interrupting you.
Yeah.
When you're going to the reduction of carbon monoxide to carbon dioxide, or oxidation, that's that's 40,000 turnover, right? In that direction.
In oxidation of CO is 40,000, yeah.
Yeah. In the other direction, which you may be speaking about, what's the turnover rate there?
Yeah, it's actually that is something like it's the best in solution are up to 150 per second. But it depends very much on mediators. And that tells us that it's probably the rate limiting step is reduction step. We don't get the electrons quick enough inside. So I guess the best guess would soon come probably from electrochemistry.
So there's a big difference in this.
They-
And obviously you're going
Yeah.
uphill thermodynamically compared to downhill. So I mean, yeah, okay, well yeah, okay. So you will talk about the actual oxidation of carbon monoxide and carbon dioxide, because that's the main reaction of this enzyme, isn't it?
It's a quickest reaction, but of course physiologically it's both. No? It depends really on what you want. No? The enzymes are the organisms growing with carbon monoxide. It wants carbon monoxide oxidation, but the ones that are growing with carbon dioxide wants to fix it.
It's scared to go in one direction. That's downhill, down the thermodynamic gradient.
If the substrate is present, it's correct. Yeah, if the substrate is present. I have to hurry because I got it. Yeah. I'm now speeding up. So okay. So this is a short summary in a way. So we know now the nickel is of course important for carbon monoxide oxidation, carbon monoxide binding, activation. Interesting here is a nickel 2+. It's not the favorite metal for binding a carbon monoxide molecule because carbon monoxide is typically binding or you get stable complexes, carbonyl complexes with nickel 0. This is a nickel 2+. But it's surrounded by pi donor ligands, making a relatively weak interaction most likely with carbon monoxide. In this way, keeping this carbon relatively void of electrons. This makes it very nicely attackable by a nucleophile, and this nucleophile is of course here activated at the iron. You have this hydroxyl ligand, which is just in line to make this attack to the carbon of carbon monoxide. So you have here the nucleophilic, here the electrophilic action, and that is relatively clear. What is unclear, and that is the big difference to the photosystem, we have no idea where our electrons are. So obviously this is 2-electron chemistry, and you would guess, of course, this cluster would pick them up, spectroscopy, Mössbauer spectroscopy, but also EPR spectroscopy relatively strongly argue against that this part takes up 2 electrons. There was an argumentation for nickel, for going to nickel 0, but that would be obvious by EXAF spectroscopy, and there's no real shift between the oxidation states. A nickel hydride has been proposed. That is still open, but the enzymes don't produce hydrogen, even when you make it more acidic. That would argue against the nickel hydride because you would evolve hydrogen. And finally, nickel-iron bond as a storage part for carrying 2 electrons were proposed. These are proposals. None of them have been experimentally verified. There's a lot of argumentation or good things against several proposals, but there's no good experimental evidence for the proposals where the 2 electrons are residing. So that's still something that we have to find out in the next years. Next decade maybe. Everything I told you so far was around the metal clusters, and of course there's more than the metal clusters. There's electron transfer, there are substrate channels, there's proton transfer. Protein matrix is important, but for the sake of time, I focused here on the metal, and with this I come to my end. I want to, of course, briefly acknowledge the people, Julia Dendra, Lilith, Jochen, who did the main work, Jayon, my collaboration partners at TU make for spectroscopy, and FU for spectroscopy, and the CNRS people for electrochemistry, and of course, finally, I want to thank you for your attention.
Thanks for a fascinating system. Fantastic. Jim.
I'm sorry, but especially can I summarize your lecture? Can I ask you these questions? I'm going to summarize. You tell me where it's wrong. This enzyme takes hydrogen out of water, 2 reducing equivalents, 2 electrons, 2 protons. The energy is coming from the oxidation of carbon monoxide to carbon dioxide. The reaction takes place on the surface, And it looks, and as far as you're concerned, I think from your previous work, it's a nucleophilic attack of onto the carbon monoxide from the water.
Yep.
This all occurs on the surface between the dangler, if I may say so, or exo ion, and the nickel within the cubane.
Yep.
And that there is no, during that reaction, there is no large structural changes of the cubane itself, the Fe3 nickel cubane. And therefore it's a sort of surface reaction. The role of the cubane here, of course, is it must be to take the 2 electrons and deliver them then into the, wherever, to a hydrogenase or into the system, because the organism is living on those electrons as well to drive its reductive chemistry as well. It's not
well
just,
summarized,
I think you give the indication that it's a means of getting rid of carbon monoxide only. This carbon monoxide is food for this organism.
It's the electrons. Actually,
it may have excess food, and it may want to get rid of the excess food through a hydrogenase and bubble the hydrogen off.
Yep.
But in many ways, in photosystem 2, we take 4 electrons and 4 protons out of 2 water molecules by a system, which a catalyst which has extremely similar geometry. And I personally feel that there's a real, as you know from my paper, but that there's a real similarity between the 2 things. And that indeed, it's highly likely that the mechanism in photosystem in PS2 is that it occurs on the surface, and it involves a nucleophilic attack between the dangular manganese and the calcium, or hydroxides on the calcium. Actually, the nucleophiles would be water on the calcium, attacking the highly electrophilic oxos associated with a high valency manganese. That's how I see it. Very, very similar. Would you agree?
In principle, I agree perfectly. I wouldn't really say that it splits water in the sense, because of course it takes the oxygen. You would principally it deprotonates it, and it takes that oxide. It takes the protons out of the water, but your principle would describe the oxygen and carbon dioxide still as an O2 minus. So formally, at least, you would say the carbon is oxidized, but the oxygen is still an oxide. It's still an O2 minus.
Hydrogen, if you like, inverted commas. That's what you put up there. The water shift reaction.
Yeah, the water gas. Yeah, then your 2 electrons would end up in
I'm not criticizing you at all.
hydrogen.
I just can see that similarity is remarkable.
Yeah. It's just I wouldn't put it so much from the perspective of the water, but more of the carbon monoxide that is oxidized to carbon dioxide.
I think we better let you convince him that that's the mechanism in the coffee break, because we're running out of time a bit here. Okay. Can we have just one last question? I'm sorry, because we're running out of time. Okay, 2 questions, and then.
Okay, very interesting. I want 2 short questions. Is this enzyme sensitive to oxygen?
Yeah, it's highly sensitive to oxygen.
Highly sensitive. Okay. The second question. So you mentioned that nobody synthesized this class.
Yeah.
Oh, why? So actually, this looks.
Yeah. So I never tried it myself, of course, and I wouldn't manage anyway. So I can only cite it from the papers of Dick Holmes' group from Harvard. And he was trying, he's certainly one of the experts with Katsuhiko Tatsumi in synthesizing iron sulfur clusters. And he said the big problem is to put a dangler that is really staying there and not saturating it completely with the ligand sphere around it. Because otherwise you would never get turnover.
Because this method is wrong.
Okay, one more very short question.
Argue with Richard Holm.
It would be very practical if we could take carbon dioxide and make methanol. You have 2 enzymes here. Could you envisage a tandem construction so you could do this?
Well, in principle, the so-called methyl branch there, carbon dioxide is taken, reduced first to formate, then this is coupled to tetrahydrofolate. That reduces it in principle to the same oxidation state as methanol. You get methyl tetrahydrofolate, which is like methanol. So the last step would just be to take the methyl group out from the tetrahydrofolate, and then you have methanol. These are, I think, 4 enzymes doing it. But then the last step should be easy.
Okay, thanks very much again, Holger, for a fantastic talk.